All right, kia ora everyone. Here is our first lesson for week number 6 of remote learning, and this is task number 1. What we're going to be looking at today is mitosis and how it relates into the body. It's really important that we review mitosis from last year. You're not really going to get a question on mitosis. The focus for this year is going to be meiosis, but it's important to talk about mitosis so that way you can understand the difference. So the success criteria from this is that you should be able to differentiate between gammatic and somatic mutations. Again, it'll tie into the meiosis material that we'll cover in the next 2 lessons. So first, before we do that, let's backtrack a little bit. So if you remember, I've divided this standard or external into 3 different chunks. The first chunk being molecular level. The second chunk is cellular level, and then the third chunk is going to be looking at the larger population level, and this is all tying in with genetic variation. So we have finished the molecular level. We looked at DNA, we looked at alleles and things like that, and now we're going to expand our scope into the cellular level. But before we do that, let's just backtrack a little bit. So when we were talking about DNA, you have to also remember that DNA is really important. It obviously is the code of life. It's basically like the computer program to explain what to do. And it's really important that this code gets into all the cells. And so for it to get into all the cells, the DNA needs to be replicated, and there needs to be cell division to ensure that all the cells get this information. Now, there are 2 different types of cellular cellular replication, also known as cellular division, and that is mitosis and meiosis. And you should have covered that last year in year 11. With mitosis, the thing in this case is that we're creating identical daughter cells. Big picture-wise, it's used for asexual reproduction, commonly in things like bacteria, but for organisms like us, we use it for growth and repair. Meiosis, on the other hand, is used in sexual reproduction, and it's producing non-identical daughter cells that are also known as gametes, which can also be known as your sex cells, so sperm and egg. Cool. So today we'll just cover mitosis, and then the next 2 lessons we'll look at meiosis. So first off, mitosis is a cellular division, and like I said earlier, it's going to produce identical daughter cells. The exception is when a mutation occurs, which is what we've talked about previously. This is really important for growth and repair, and this happens with our somatic cells. Somatic cells are also known as your body cells. This is also used as an asexual form of reproduction, and you're basically, like I said, making identical copies of the original. With this cellular replication method, because they're exactly the same as the parent cell, we're also going to see the same number of chromosomes as the parent and the same DNA. So that, again, is an important layer to think about as a compare and contrast. Body cells are also known as being diploid, because that means that they have 2 sets of chromosomes. What else did I want to add about that? With this mutation material, about how it's going to be genetically identical with the exception of mutations, these mutations are really important in a sense of context. If you have a mutation in a somatic or body cell, it only affects the offspring, or I should say the daughter cells of that. This can't be passed genetically down to the offspring. It'll only affect the individual. The example of a mutation in a somatic cell is cancer. And so when you have one cancer cell, it divides, it replicates, and that's how you can get a cancerous mass. Anyway, let's kind of look at that link between what we've talked at the molecular level versus a cellular level. So molecular level, we've been talking about DNA and how it's this double helix. Now, if this was all completely unwound in our cell, it's obviously going to be a huge mess, which is fine. We want it to be unwound so that way we can have access to the genes. However, during cellular replication, we want it to be nice and tidy and easy for the cells to divvy it up. And so what we see then is that this DNA gets wrapped around histones, and then that keeps getting wrapped around and wrapped around and wrapped around until you have the chromosome that's being formed. So I'm trying to think what else I wanted to say about that. The analogy I like to use between chromosome and DNA is, think of DNA like a string of yarn, and then a chromosome is a ball of yarn. It's much easier to divide up piles of balls of yarn than completely unwound yarn. Cool. So some more things about chromosomes. Like I said earlier, these are known as diploid cells because you have two sets of chromosomes. You get one from mom and you get one from dad. Humans have 23 pairs, so 23 times 2 is 46 chromosomes. You can see this right here. However, with the sex cells, we have half that amount. So in the sperm or in the egg, there would only be 23 chromosomes. The reason why it needs to be halved is because when fertilization occurs, you're going to get a restoration of the chromosome number. If we didn't do that and we didn't have meiosis, we'd be doubling our chromosome numbers with each generation, and that can cause a lot of problems. I think it's just really interesting to see the just the number of chromosomes with each different organism. And I'm amazed that there's so many in like kiwi fruit and yams, or I think that's a yam, compared to us. So just because you have a lot of chromosomes doesn't mean you're a more complex organism. So different species, different number of chromosomes. Pretty cool to look at. Cool. So let's now move on to the process of how this happens. I'll switch the document camera to show you these steps, and I think I pause somewhere in the middle of it to show you some more of the terminology. I'm happy at this point if you guys want to pause the video and watch the Amoeba Sisters video that I linked on my Google site. You might find that a bit easier to follow because it's an animation, whereas I'm just going to be writing the notes down. What else did I want to add about this? I think that's it that I wanted to talk about. So let's go ahead and look at the notes. Now I remember what I was going to talk about. Talk about when we actually do our drawings. Yeah, sorry about the shadows. All right, so before our cell goes into mitosis, we have a stage called pre-mitosis. This is just the cell kind of chilling and hanging out. I like to keep track of the cells and the DNA amount because then you can see the process of the doubling and then the dividing. It's also really useful because when we compare it to meiosis, you'll see what I mean. I think that's the main thing I want to talk about. There's obviously some vocabulary words with the steps of the cellular division. You're not expected to know that. It's just to give context because you're going to probably see it pop up if you're looking at any other resources. So yeah, keeping track of the cells, keeping track of the DNA. So if I have pre-mitosis, I only have one cell. And what I'll do is I'll draw the nucleus, which is that black circle. And with that nucleus, I'm going to go start doing DNA colors. And I'm going to do it with two different colors just to represent the two different sets. So the DNA is completely unwound and accessible in the cell during pre-meiosis because we want to be able to access our genes. With the DNA, there's two sets of it, and I'm going to write 2X to represent that you have the diploid. I'll also write 46 chromosomes. That's just specific to humans, so that way you have an example that's working. So that's humans. All right, cool. So when our cell then enters mitosis, the first thing is that it's going to replicate the DNA. So with that replication process, we are basically doubling the amount of DNA that's possible. So I'm going to draw that as basically more squiggly lines. So that way it looks more dense inside the cell. DNA replication, the steps of that is not expected to be known in this standard, but if you want to pick up the external, I can't remember if it's cellular processes is what it's called, or the other genetics one, but one of those two the steps of DNA replication is one of the things you need to know. So you can find that in your SciPad if you're interested in knowing the specifics on how that DNA replicates. And in that process of DNA replication, the mutations can occur. Right now, I still have one cell, but because I've replicated my DNA, I have twice as much DNA possible. So in theory, if I was thinking about chromosome amount, I could do 46 times 2, which is 92. They're actually as like a number to help you kind of think about what's happening. Cool. All right. After the DNA replicates, we obviously want to concentrate it down into chromosomes so that way it's less messy. But before that happens, the nucleus starts to degrade and break down. So that way we can actually start pulling and dividing later. And the DNA starts to concentrate. So when it starts to concentrate, it's going to make chromosomes when it does that. So I'm just going to draw a few. Obviously, I'm not going to draw 92 chromosomes in there. Or to say 92. I'm not going to draw 46. I'll show you what I mean by 46 and 92. All right. So cell-wise, we have one cell. We have 4x 92. That's what I'm thinking about. So let me move back to the slides because there's important terminology that I need to refer to that can get kind of confusing. Cool. So the word chromosome is used twice in two different contexts, which makes it confusing and frustrating. Both of these things are chromosomes. The way I like to distinguish them is this is an unduplicated chromosome. So basically, when we were looking at pre-mitosis, if it had condensed, it would just look like that. You are more commonly going to see chromosomes as X shapes because whenever we take photographs of cells, we're taking the photographs during mitosis when the chromosomes actually form and that DNA concentrates. And so that's why you commonly see the X pattern when it comes to chromosomes. And the terminology is correct in saying that's a chromosome, but this is a duplicated version of chromosome. The other important terminology is that this half of a duplicated chromosome is known as a chromatid. And when we start looking at the splitting in mitosis, we'll see those sister chromatids split apart, and then you use the word chromosome still. It's very strange. I'm very sorry, whoever decided to use that terminology. The other thing is that centromere, and that's what's holding your sister chromatids together. So, as I was saying, the drawing I made obviously has the X versions because my DNA has replicated. Next step is that my chromosomes need to line up in the middle. And they're going to do that in a single file line. So I'm just going to draw all 4 in the nice center. And I'm trying to draw them different sizes, and hopefully I can fit them all in. So we have one cell, double the amount of chromosomes that a normal body cell would have. And these guys are able to move into the center because these little spindle fibers are basically shifting them around to move. I think those are called centrioles, the ones that make those fibers. So yeah, they line up in the center, and the whole idea is when they line up in the center, the little spindles will pull the sister chromatids apart. So we're going to have that pulling apart of the duplicated chromosome. Like I said, this is where the terminology gets confusing because when they split apart like this, these guys are referred to as chromosomes still, even though they split in half. So chromatid, chromatid, chromosome, chromosome. Sister chromatids pull apart, chromosome, chromosome. So that's where, when I say there's 92 chromosomes, 1, 2, 3, 4, 5, 6, 7, 8, that's what I'm referring to. It's just a number to kind of show you idea-wise what's happening. Anyway, I still have only one cell at this point. and I still have more DNA than I need. But what's going to happen is those chromosomes are going to split apart, and my cellular membrane is going to start forming and basically pinching off and pulling apart. And eventually that pinch apart will form two separate cells. So then what happens is my nucleus forms again, my DNA starts to unravel. Obviously, I don't have it as dense as this one because I've split the DNA in half. And again, those chromosomes make it a lot easier to split it in half. So when I then look at the final step, number of cells that I now have are 2 cells. Each cell now is back to that diploid number, which is a total of 46 chromosomes. And are these cells identical? Yes, they are. And like I said, I want them to be identical for growth and repair. Don't worry, I will scan it. Cool. All right, the next thing I wanted to show you is some videos just so you can see mitosis in real life. So these are taken with microscopes. They've obviously used fluorescent dye so you can see the chromosomes. And like I said, this is the reason why we think of chromosomes as the X shape, because you only see them condensed when mitosis or meiosis is happening. Anyway, I think it's just really neat to see it. And you see how they pull them apart, make 2 cells. I'm not sure why that looks like 3 cells. It should only really be 2 cells. So I'm not sure what happened in that video. Or in that cell. Anyway, yeah, you can see that pinching of that, the membrane. And I think they're just more of the same. Cool. All right, and here's another one that you can watch. I'm just gonna mute it. So I can talk you through what's happening. This one's a little bit in a slower motion, but you can see those chromosomes. And so this is it trying to arrange itself. Obviously, they sped up the video because this is actually quite a long process. And there you go, the splitting. So there's the sister chromatids splitting up, and you can see in the center here, that's the cell membrane pinching, so it forms 2 new cells. And here's some different versions with different dyes. It's just really pretty to look at. There you go, some more examples. Anyway, I'm gonna go ahead and move on. You can watch the full video on my Google site, but let's do a quick little recap. So some things to note as kind of important things to remember with mitosis is, let me just grab a marker. So the first thing to think about is, where does it occur? So this occurs everywhere in the body with the exception of the sex organs. So when I say sex organs, I'm talking about the testes and the ovaries. Number of divisions, there's only 1. Again, important to note these things because when we do meiosis, you'll see the difference. Type of cell it creates is body cells, also known as somatic cells. Number of chromosomes stays the same. Number of cells produced is 2, and the role in the body is for growth and repair. So that's mitosis. All right, cool. Some other final things to note. I won't play this video, but if you want, it is on the Google site, and it's a really neat video to watch because this goes from zygote, which is a single cell just after fertilization, sperm and egg come together, a new a new organism, all the way to tadpole. So you can actually see how that one cell divides into an organism. So it's a really cool, neat video to see. It's not like the other ones, which were more focused on the chromosomes. This is focused on mitosis as a process to get from one cell to organism. All right, cool. So there's some considering the following questions. First off is a true or false. So we obviously have different cells in our body, say, for example, skin cells and muscle cells. Very different functions, very different structures. The true or false was, do you think it has the same DNA? So take a moment to think. 50-50 chance. The answer is true. All of these different cells have the same DNA, which is really neat to think about. It's a common misconception that different cells in the body have different DNA and that's what makes them different cells. That's not what happens. What happens is different genes get expressed in each cell, or cell type, I should say, and that's why it gives it those specific properties. That's why, again, why we have 23 pairs of chromosomes, because there's lots of different DNA in there that's going to be controlling for different traits that need to be turned on and off. In fact, there's been some really crazy kind of mutations or like weird interactions with DNA. And I've seen and heard some really crazy things where like teeth will start growing in people's muscles because the teeth genes got activated by accident somehow. It's really nasty stuff. I don't suggest you Google images for that. But yeah, that's kind of the idea behind it. That's also the reason why we can't just take a skin cell and kind of reverse it and make a liver cell quite yet, because we need to be able to turn on and off the genes to revert it backwards. It's a cool area of science that we're working on. Oh, I answered that question. How is that possible? It has to do with how the DNA is wrapped. So the genes that we don't need to access for that type of cell gets wrapped around the histones, and they're inaccessible for any gene expression. If you want to understand that more, you need to do the external on gene expression. And that'll actually explain how do we go DNA to the trait, which we briefly covered, but it obviously goes into more details. There is a little asterisk with the red blood cells. Red blood cells actually have no DNA. They've removed the nucleus and basically all the different organelles inside the red blood cells, so that way you can pack it full of hemoglobin and carry lots of oxygen around. It's also a good thing that it doesn't have DNA in it, because it means that we can do blood transplants and blood transfusions without any issue, because there's no DNA in there. Cool. All right, so that is today's lesson. You were exploring mitosis and relating it to the body. There's not necessarily going to be real specific questions on it when it comes to this external, but I needed to revise it because it gives better context than for meiosis. Cool. So go ahead and go on to my Google site and do your Mahi choice. And like I said, all resources are on Google Classroom.